Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Long, and I'm going to be doing some complex information in this lecture. Before I get into it, know that we're in the coronavirus shutdown. These videos are being done quickly and crudely and in an impromptu fashion from that little kitchen or dining area in my home. So if you hear background noise, it might be kids microwaving something or my dog bark. I apologize. I have no other options. So today's lecture is going to be intended for students taking my Biology 2402 class. And so I'm going to deliver this lecture in a manner which is designed for my students. And if you have another instructor, either at Del Mar College or another institution, this may not cover all the details that your instructor wants to go over. I'm going to simplify some things because I realize that my students do not have a lot of chemistry in their background. Some of them are currently taking chemistry, some of them are taking chemistry next semester, and so some of these concepts would be very difficult to understand if you don't have the basic background. And so I'm going to set that background some. I'm going to simplify things. I could make this far more complex. I have a very profound understanding of a lot of this stuff. My master's was done in a protein biochemistry lab. It was in neurobiology, but we were doing a whole lot of complex protein chemistry. And so I understand this stuff at a very high level, but I'm going to break it down for that introductory level student taking their first human A&P courses so that you can understand this. So for those of you out there that want to criticize or critique what I'm doing, if it's too watered down, I don't really care. This is for my students. Now, if you're going to follow along in my note set, there's nowhere to do these first part of the video. This is the setup so that we can get into the note set and we can understand what's going on. So now, when we look at atoms, the periodic table of the elements shows you all the elements, and an atom is one unit of each element. And when we look at all atoms, one of the things you need to know, or should know, is that all atoms are made up of three subatomic particles. There's one called a proton that has a positive charge. There's one called a neutron that has no electrical charge, hence the little zero. And then there's the electron. We covered some of this in part 1 A&P, but I need to step back and do this. Now, if I'm going to form an atom, I need some number of protons, neutrons, and electrons. The only exception is hydrogen has no neutrons. A hydrogen atom is nothing more than a proton in the nucleus, and then the electron, a single electron whizzing around it in its electron cloud or orbital or shell. There's different terms used. So every time I make an atom, I need some number of protons. The protons and the neutrons hang out kind of very densely packed in a nucleus, and then the electrons are whizzing around in this area around the nucleus that we call orbitals, okay? And if you recall early on, we had what was called the 288 rule for some of the more simple atoms. This is the number of electrons that actually can share the same orbital. And so the first orbital will have 2 electrons, can hold up to 2 electrons. If we have any more, then we have to add more electrons, but we have to create another orbital. For example, let's say we pick an atom that has 8 protons, okay? It's going to have 8 electrons because atoms are electrically balanced, but only 2 of them can fit in the fourth. Let's do this one, 6 protons, okay? There's 2 electrons here, and then I got to fit 4 more electrons in the next orbital. I could fit up to 8. Once I get beyond 8, I have to create a new orbital. Now, we covered some of that stuff early on in part 1, and I'm not really concerned a whole lot about all of this stuff. I'm not really going to test you on that any longer, but what I need you to know is that atoms are made up of subatomic particles, and we're going to focus on hydrogen for a reason in just a moment. Now, it's hard for people to understand the concept of energy because those subatomic particles, the protons, the electrons, and the neutrons, they are nothing more than little condensations, so to speak, of energy. This isn't the best way to explain it without doing a lot of complex physics, though. I got to get it across to you in a simpler way. One of the things that we can relate to, especially down here in South Texas, is humidity. Humidity is the amount of moisture that's in the air floating around us. And as you know, there are water molecules floating in the air out here, And the more humid it is, the more water is in the air and the more densely packed it becomes. Now, if we look at a molecule of water, it's made up of oxygen and 2 hydrogens bound together in some covalent bonds. But because of the extra electrons on the oxygen, the electrons give the oxygen more of a negative charge, and because of the pull of the electrons in this direction, it makes the hydrogen somewhat positively charged. Okay. Now we can go into the reasoning behind that. It's not important. But that gives a polarity, a negative and a positive polarity to a hydrogen molecule, to a water molecule. So now, if another water molecule gets within a close enough proximity, the negatively charged oxygen can form what's called a hydrogen bond with the hydrogens of another water molecule. And what that would allow us to do now is put a whole bunch of water molecules next to each other with all these hydrogen bonds. The hydrogen bonds are not very strong, and they allow the molecules to kind of wobble and wiggle around each other. And if I do this with enough of these water molecules, if I continue to bring some more and more close, because of their electrical charges or their polarity, they will start to stick together. Now, if I keep them far apart, they'll whiz around the universe and never come together and form a bond. But if I get 2 of them close enough to each other, they can form one of these hydrogen bonds and kind of hang out together and stick together. Now, if enough of them coalesce together, their mass becomes so much greater than all the other air molecules, all the nitrogen and oxygen in the atmosphere can support. And eventually, it will precipitate out of the universe as a drop of water. That's why you see dew on your windshield in the mornings, or why you feel the water drops when it begins to sprinkle or rain. So, excuse me. Sorry, I had to sneeze. My allergies are going crazy right now with all these mesquite trees. So, now the reason I explain this to you is I don't really care about the water, but energy works sort of in a similar way. There are little subatomic particles. Sorry, I'm having a sneezing fit. There are subatomic particles that have electrical charges that are whizzing around the universe like this, protons and electrons and neutrons. And if I bring 2 of them close enough together, then the electron will whiz around the nucleus by some electrical forces, by some forces that hold them together and pull it together. And if I start to make more and more and more of these, they'll form atoms. And then atoms will combine in different ways to form molecules. Molecules will combine to form organelles, and then cells and tissues and organs and organ systems, all the levels of organization. So what I need you to understand is not so much water, but that these subatomic particles, electrons and protons, are kind of like that in the sense that if they come close enough, there are some electrochemical forces that hold them together, and then as they're held together, they can grow and grow and grow and grow into molecules. So subatomic particles form atoms, atoms form molecules, molecules can form other structures. Now, that's going to be an important concept for us in a little bit. So all hydrogen atoms and all subatomic particles are little condensations of energy, like a condensation of water, so to speak. Now, before I go into the actual chemistry that we're going to talk about, I want to cover one thing that's on page 86 of my notes set. There are some definitions that you guys need to know. So I'm going to write these out with you, and we're going to talk about them. Or I may not write them all out, but I'm going to talk about them. Metabolism is usually defined as the sum total of all biochemical reactions. And when we're talking about human anatomy in our bodies, or in whatever organism you're talking about, that would be their metabolism. So in our metabolism, we're talking about all the chemical reactions combined. Now, what we're going to focus on is called energy metabolism. So it's a subcategory of metabolism, but that's what metabolism means. You should know that definition. Now, metabolism involves 2 different types of basic chemical reactions. Some of them are called anabolism or anabolism. So ana means up, or it can mean with. So like putting things together. So I like to define these as what we call build-up reactions or building reactions. Now, in chemistry, you would call these synthesis reactions. And in a synthesis reaction, what you do is you're synthesizing larger molecules from smaller ones. So in an anabolic reaction, in anabolism, this is the building or synthesis of large molecules. Sorry, my marker is squeaking. I'm going to see if I can change it out, but I want to get a color that shows up very well. Of large molecules from smaller ones. Okay, so if I have compound A shaped like such, and I have another compound that's shaped something like this, B, and these two pieces can fit together. I didn't do that very well. And they go together and form a much larger compound, C, that would be a synthesis reaction. Like building amino acids into proteins is an anabolic reaction, building up, getting bigger. Now, the opposite of that is going to be a catabolic reaction. Catabolic reactions would be the exact opposite, which is where I would actually take a large molecule and break it into smaller ones. The kata comes from an Asian word, kata, which means to cut. So in a catabolic reaction, or in catabolism, in chemistry, we often refer to these as degradation reactions. To degrade means to break down. And so this is the breaking of large molecules into smaller ones. Okay? We are literally breaking stuff apart into smaller molecules. They're digestion reactions also, like the breaking down of carbohydrates into simple sugars, or the breaking down of proteins into amino acids. Now, let me get into two other definitions here that are in the list, and then we're going to go into the chemistry. Okay? This video is going to run about probably 25 minutes or so. I apologize. I'm trying to keep them short, but I just can't. There's a lot of information we have to cover. So there's a term called a catalyst. A catalyst is any substance that increases the rate of a chemical reaction. So it doesn't matter if it's an anabolic or a catabolic reaction, any chemical reaction. So let's say I have substance A plus substance B that are going to combine in a catabolic reaction to form substance C. Well, we can measure the amount of time it takes for that chemical reaction. For example, two amino acids can form a peptide bond, but in nature, that might take, I don't know, 1,000 hours. If I put them in, that's a long time. That's not fast enough for life to occur. Now, what if I took substance, I don't know, let's just call it substance NZ, which is actually an abbreviation for the word enzyme. Okay, but let's just say I take some other substance and I put it in here. And when I mix that with the other compounds, this goes to 0.0000019895 seconds. It happens almost instantaneously. And this substance can be pulled back out unaltered. Then we would call that a catalyst because it reduced the time that it took for the chemical reaction to occur. That's all that they do. They increase the rate of chemical reactions. Or some people say in chemistry, it lowers the energy of activation. We're not going to get into that. You'll learn all that in chemistry. I just want you to think of it in these terms. Now, if these were biochemical, organic molecules, then this would be an enzyme. So the definition of an enzyme is the exact same definition as a catalyst. We change one word. An enzyme is any substance that increases the rate of a biochemical reaction. We just changed the word chemical to biochemical. Enzymes increase the rate of biochemical reactions. They are catalysts. They're biological catalysts. They're almost always in nature, 99.999% of the time, they're proteins. Proteins fall under a number of classes of proteins, and one class of proteins are called enzymes, and enzymes are proteins that increase the rate of biochemical reactions. They make the biochemical reactions happen much faster. Got it? All right. So now, in chemistry, if we were to look at this, we would call these molecules over here the reactants, or these substances would be called the reactants because they're going to react, and this would be called a product. Okay? But in a biochemical reaction, we don't use the term reactants, we use the term substrate. The substrate is any substance acted on by an enzyme. So if we're going to combine two amino acids into a dipeptide, then the amino acids would be the substrates, the product would be the dipeptide. So in biochemistry, we call the reactants substrates. Essentially, it's the same definition. Okay, it's any substance acted on by an enzyme. Now, the active site is the region on the enzyme where the catalytic or the enzyme activity takes place. So let's say I have two substances shaped like this. I have substance A and I have substance B. They have some three-dimensional shape, right? And for some reason, I'm going to change our shape a little bit. I can't get them to go together very easily. Now, there might be another part here where they will interact. But I got to get them in just the right orientation for those molecules to form a bond, or those two substances. Well, if I have my enzyme down here, what happens is the enzyme might have an area that each one of these can bind to and stick, and then they'll form their chemical reaction. They'll form their bond. This area here would be called the active site. The active site is the location or the region on an enzyme where the enzyme activity or catalytic activity occurs. Got it? I'm not going to write that out for you, but there it is. So those are some of the definitions I want you to know. You need to know the definitions of metabolism, catabolism and anabolism, substrate, enzyme, catalyst, and active site. Those terms will be used a lot in your future if you do any more chemistry. Now, let's get on to energy metabolism. Before I go into energy metabolism, I'm going to do one more thing, and then we're going to stop this video, and I'm actually going to do a second metabolism video. I'm kind of improvising as I go along, because I don't want this to go on too long, I think. So, when it comes to energy metabolism, what we really focus on is the production of ATP. As you know, that third phosphate, when we break that bond, it releases some of the energy combined in that covalent bond. It takes energy to form a bond, and then when we break it, some of the energy that went into it gets released. Now, in order to make ATP, we need a couple of substances, and in order to make enough ATP. So one of the basic chemical reactions, and I've drawn this a lot in my classes, and I'm going to continue to do it. I'm going to simplify it first. We'll get a little bit more complex. Okay. What we need is a molecule called glucose. I'm going to make it a G with a circle around it. That's glucose, right? Glucose plus oxygen, O2, in the presence of mitochondria. The reason we need the mitochondria is that they contain a lot of the enzymes that are necessary to make these chemical reactions happen. It's going to convert this into 36 ATP. There's the energy we need, a lot of it, plus some carbon dioxide, plus some water. That's some of the waste molecule that we have to exhale when we exhale. Okay, so that's a real simple way of looking at this equation. Okay, now we're going to get a little bit more complex. So when we look at glucose, and there's two other simple sugars, fructose and galactose, that have the exact same chemical formula. It's the order and the arrangement of the carbons, hydrogens, and oxygens that make those three simple sugars different from each other. But we can use glucose, fructose, or galactose for these chemical reactions. We tend to focus on glucose. It's the most efficient, it seems. So the chemical formula for glucose is H12O6. These little subscripts tell you how many. There's 6 carbons, 12 hydrogens, and 6 oxygens in one glucose molecule. Okay. Now, if I combine that with oxygen, and we actually get 6 of these, okay, we can do a balancing of the equation. I'm trying to make that C look real pretty, but I'm kind of too close to the board. I'm having to slide around here. So if I take 6 glucose molecules in the presence of 6 oxygen molecules, I combine them with mitochondria. I'm just going to put mito up here for mitochondria. That's where I'm going to get my 36 ATP. Plus I'll get 6 carbon dioxides, plus I'll get 6 waters. We have to balance our equation. I have, it's not 6 glucoses, my bad. 1 glucose plus 6 oxygens, sorry. Now if I look on this side, I have 6 carbons. Over there I have 6 carbons. Here I have 12 hydrogens. There I got 6 times 2, that's 12 hydrogens. I got 6 oxygens here. And I got 12 oxygens there, so I have to have 18 oxygens. Well, there's 12, 6 times 2, and then 6 more, 18. So it's balanced. This is the balanced equation for this, okay? Now this is what I want to focus on. The reason I do this is because I want you to see that glucose and the 3 simple sugars that we use for energy are all made out of carbon, hydrogen, and oxygen. Now, let's take a step back. I want you to understand this because this is important sort of for our understanding of some things that we've already done and we can continue to do and talk about. We had 4 major classes of organic compounds. We talked about carbs or carbohydrates, large masses of simple sugars like glucose, fructose, and galactose bound together in these 3-dimensional arrangements. Okay? When we digest those, we release the simple sugars. We digest them with enzymes, and we get simple sugars for energy. So carbohydrates directly are used for energy production. The second major organic compound that we talked about was lipids. We usually talk about proteins next, but I'm going to talk about lipids. Lipids are made out of carbon, hydrogen, and oxygen. Okay? One of the lipids we talk about are called the triglycerides. We use triglycerides for energy sometimes, and when we break them down, when we digest them, we get 2 things out of them. We get a fatty acid. Fatty acids are made out of carbon, hydrogen, and oxygen. Lots of carbon, hydrogen, oxygen. They're pretty big molecules. And then we get glycerol. And if I look at the chemical formula for glycerol, it's C3H8O3. Well, I'm kind of building up towards that. I need a couple of more carbons and oxygens and hydrogens. And fatty acids, if I digest them, I can cut some of these larger molecules down into glucose, and some of the leftovers I can combine in some enzyme reactions. And I'm not going to get into all the chemistry because it's rather complex, but essentially, if I have to, I can convert these 2 into glucose for energy. I would prefer to use my lipids for cell membranes and growth and repair, but if I'm out of sugar, I don't want to die, so I can convert fatty acids and glycerol and other lipids into energy. Okay. Now, the third major class of organic compounds we talked about, called proteins. When we digest proteins, what we get out of them are these molecules called amino acids. Okay. Now, again, I would prefer to use amino acids to rebuild the proteins in my body that are being broken down or for growth and repair and other things. But if I look at the structure of an amino acid, our body and most proteins in nature use the exact same 20 amino acids. There's 20 of them that we use for proteins. All of them have a similar structure. They have a centrally located carbon. Now, the chemistry and the physics of a carbon molecule are such that it can have up to 4 chemical bonds. And on one side, we're going to have this group that's an H2N. That is where the amino in amino acids called. Anytime I have a nitrogen with 2 hydrogens, in chemistry that's called an amino group. On the other side, I'm going to have a carbon with a double bond oxygen and then a hydroxyl group. And this is what makes this as an acid because, as you know, acids are hydrogen ion donors. So if I pop that hydrogen ion off, then I get a negative. So This is the acid part. So amino acids all have on one side of their central carbon an amino group and an acid group on the other side. Now, over here is what we will call an R group. The R group can vary. It's carbons and hydrogens and oxygens. In some amino acids, it's simply one more hydrogen. In others, it's a methyl group or ethyl groups and some other things, which means something to a biochemistry. And then there's another R group over here. And those are carbons, hydrogens, and oxygens. In two amino acids, there's some sulfur in there, but we're not going to talk about that. So now look. Essentially, this entire molecule is carbon, hydrogen, and oxygen, except for the amino group. Now, I want to use this and stick them together to form new proteins. But if I'm out of energy, if I've burned up all my sugar, I've burned up all my fat, rather than die, our body will start to digest muscle and proteins. It'll break down protein and convert it into energy. One of the big deals in this process, though, is we have to remove the amino group. And we say that is to deaminate. To deaminate simply means I'm going to rip that amino group off. I'm going to remove my amino group. And everything that's left over is carbon, hydrogen, and oxygen. With some more chemistry, some more reactions, I can convert that into glucose. And this is what we talked about when we were talking about the effects of certain hormones. We talked about glucagon, and then we talked about cortisone or hydrocortisone. They do gluconeogenesis. They convert fats and proteins into sugar for energy. This is gluconeogenesis. One of the problems is we have to remove the amino group, and if I combine this amino group with another hydrogen, and oops, I put a plus on there, it's supposed to be a negative. What I end up with is ammonia. As you know, ammonia is a toxin. Ammonia is poison. If you drink enough ammonia, it will kill you. I'm sorry, let me let my dog out. Come on, Bear. I apologize for the interruption, but I'm not going to kill a 20-minute video, 25-minute video for a barking dog. So let me just let him out. So now, all three of these major organic compounds can be converted into sugar. That's the big picture here. These are made out of carbon, hydrogen, and oxygen, go directly into energy metabolism. Lipids are made out of carbon, hydrogen, and oxygen. I need to manipulate them and do some chemistry, but eventually, if I need to, I can convert them into sugar. Same thing with amino acids. I have to deaminate, and this toxin is dangerous. If it builds up in your body, it will poison you, which leads to the urinary system and formation of urine. That's going to be our segue to the urinary system. And then finally, the last of the organic compounds we talked about were nucleic acids. Now, technically, we can combine or break down the nucleic acids to form energy. When I break down nucleic acids, I get the three components of the nucleotides. I get a 5-carbon sugar. Let's say it's C5H10O5. Well, I'm just a few carbons and hydrogens and oxygens away. So technically, this substance can be kicked all the way over here. I know it's getting messy. I don't expect you to know all of the directions here. But I can run that with some enzyme reactions and combine it with some other carbons, hydrogens, and oxygens and form glucose. One of the other things I get out of here is the phosphate. That PO4 minus is this phosphate. And I get another substance called a nitrogenous base. Adenine, guanine, cytosine, and thymine, or uracil. Well, this has nitrogen. It's made out of carbon, hydrogen, oxygen, and nitrogen. So essentially, on all of these, I have to deaminate and remove an amino group, and I'm left over with all of these carbons, hydrogens, and oxygens that I can convert into sugar. So now, technically, biochemically speaking, I want to use carbohydrates to break down into simple sugars for energy metabolism. I can use lipids and break them down into fatty acids and glycerol and convert that to energy under gluconeogenesis. Proteins can be broken down into amino acids. I have to deaminate an amino acid, and I'm left with carbon, hydrogen, and oxygen. I can use that for energy, gluconeogenesis. This is when you're in deep starvation and you're in deep trouble. So if you watch TV shows like Alone or Naked and Afraid, where people are losing 25 pounds in 21 days, they're breaking down all their muscle. All their fat disappears, and then they break down all their muscle. If you have to get to nucleic acid breakdown, you're pretty much dead. You're not going to survive. So we really don't get too much into it. Now, the whole point of this is just to reiterate what we were talking about before when we talk about the 4 major organic compounds. And what you need to know is this. These are made out of carbon, hydrogen, and oxygen directly for energy. These are made out of carbon, hydrogen, and oxygen. I would rather use them for membranes, but if I have to, I can convert them to energy. The big deal here is the proteins. I want to use them to build proteins, but if I'm out of every other energy source and I can't chase down a rabbit or find some kelp or something to eat, then I can convert amino acids under gluconeogenesis into glucose. But I have to deaminate, and that's where we're going to generate some very toxic compounds, a lot of amino and ammonia containing compounds and ammonium ions. And that's going to be the necessity of the urinary system and why we're linking it to metabolism. And of course, we can break down nucleic acids for the same process. What you need to know from this diagram is simply this. All of these can be used for energy if necessary. These two I have to deaminate and form some ammonia-containing compounds, which are dangerous. But all of them can go from C6H12O6 in the presence of 6 oxygens in mitochondria. I get 36 ATP, a ton of ATP from 1 molecule of glucose. It's almost like getting 36 miles to a gallon in your car. Now, we can convert glucose into energy without oxygen and without mitochondria, but it only nets us 2 ATP. That's not enough. If you only got 2 miles per gallon, you wouldn't even make it from one city to the next on a road trip before you would burn up all the energy and the car would die. So that's good for bacteria and some simple organisms, but our bodies require, we burn so much energy, we require this complex process to maximize the amount of energy production from 1 molecule of glucose. That's going to lead us into the next lecture, so I'm going to stop this here, and I'm going to start the next lecture, and most of your test questions are really going to come from the definitions that we did, and then from this equation, and then from the next lecture that we're going to do. This is sort of a setup. I hope you had as much fun as I did. I hope you learned something. I hope it made some sense to you. And I hope to see you on the flip side in the next video. All right? Thanks for watching.